Hello dear friends, welcome to another session. In this session, we will discuss the transcription in prokaryotes, and we will specially deal with the MCQs, quiz questions with reasonable answers. Let's discuss the enzymatic synthesis of RNA from DNA is known as. The answer is transcription. The synthesis of RNA is transcription. Synthesis of DNA is replication, synthesis of proteins is translation, and the synthesis of DNA from RNA is known as reverse transcription. Next question. The transcription is catalyzed by the enzyme. The answer is RNA polymerase. The DNA polymerase catalyzes the synthesis of DNA. Reverse transcriptase catalyzes the synthesis of DNA from RNA. Next, the following is true in case of transcription. Answer: all. It requires template, it do not need primer, and the new strand is synthesized from 5' to 3' direction. Next question. How many types of RNA polymerases are present in prokaryotes? Answer is 1. Only one type of RNA polymerase is present in prokaryotes. Next, how many kinds of RNA polymerases are present in eukaryotes? Answer is 3. 3 types of RNA polymerase to synthesize 3 types of RNAs. Next, name the type of RNAs synthesized by prokaryotic RNA polymerase. The answer is all. The single type of RNA polymerase in prokaryotes can synthesize mRNA, tRNA as well as rRNA. Next, name the subunits present in prokaryotic RNA polymerase. Answer is all. It has 2 identical alpha subunits, beta, beta prime, sigma and omega subunits are present in prokaryotic RNA polymerase. Next, the RNA polymerase with all the subunits is known as. The answer is holoenzyme. The holoenzyme contains all the subunits, 2 alpha, beta, beta prime, sigma and omega. The RNA polymerase without sigma subunit is known as. The answer is coenzyme. That means the holoenzyme minus sigma is nothing but coenzyme. The alpha subunit is encoded by the gene. The answer is RPOA gene. Alpha subunit is encoded by RPOA gene. Next, beta subunit is encoded by, answer is RPOB gene. Likewise, beta prime subunit is encoded by RPOC gene. Sigma subunit is encoded by RPOD gene. The alpha subunit acts as, the function of alpha subunit is binding to the promoter binding. Next, the beta subunit acts as the catalytic site and binds to NTPs. It catalyzes the reaction. Next, the beta prime subunit acts as DNA template binding. It binds to the DNA template and catalyzes the reaction. The sigma subunit acts as, answer is recognition of promoters. It recognizes the promoters present on the upstream in prokaryotes. The sigma subunit is essential for the recognition of promoters. The subunit inhibited by rifampicin is. Rifampicin is an antibiotic. It inhibits beta subunit. The antibiotic rifampicin inhibits transcription at, the answer is initiation. it inhibits only initiation, but it cannot inhibit elongation termination processes. RNA polymerase holoenzyme is necessary for which step? Answer is initiation. For the initiation of transcription, RNA polymerase holoenzyme is very much essential. Next, RNA polymerase core enzyme is necessary for which process? Elongation as well as termination. Next, the polymerization rate of prokaryotic RNA polymerase is, it is 50 nucleotides per second. Next question, the sequence of -10 promoter in E. coli is TATAAT. It is also known as a Tata box in general. The Tata box names in prokaryotes and eukaryotes. There are different names for Tata box in pro and eukaryotes. In prokaryotes, it is known as a Pribnov box, where it is present at -10 region. In eukaryotes, the Tata box is known as Hogness box, which is present at -25 region. The sequence of -35 promoter in E. coli is, it is TTGACA sequence. Other names for template strand are, the template is also known as antisense strand, non-coding strand, or minus strand. The other names for non-template strand are sense strand, coding strand, or plus strand. The first nucleotide that is added during the transcription is, answer is either ATP or GTP. The speciality of this nucleotide is, from the first nucleotide, the pyrophosphate is not removed. That means the 3 phosphates are intact for the first added nucleotide. Next, what is open promoter complex? The open promoter complex is formed when both the promoters at -10 region and -35 are recognized by the RNA polymerase holoenzyme. Suppose if this is the RNA polymerase holoenzyme, and if it recognizes both the promoters at -10 region and -35 region, then it forms the open promoter complex, where the local unwinding of the DNA takes place. Next question, which subunit comes out after the addition of 8 to 15 ribonucleotides during initiation? After forming 8 to 15 ribonucleotides, the sigma subunit dissociates from the RNA polymerase. It forms the core enzyme. Then the core enzyme is responsible for the elongation and termination. Termination of transcription may be, the answer is, it may be either row dependent termination or row independent termination. Thanks for watching till the end. Do watch other videos in relation to this video. If you like this content, please comment, share and subscribe. Thank you.